Now let's go through the protocol for endospore staining. So endospore staining is very similar in the process to gram staining. So for this lab, we're going to fix a slide of the bacteria Bacillus subtilis and do a very similar process to what we did for gram staining. We use Bacillus subtilis in this lab because it's an endospore former. So you should see in the end a slide where the endospores are stained green and the vegetative cells are stained pink. So to do this lab, you start with a plate with colonies and a blank slide. And we also have our slide staining tray here. And the first step is to use a wax pencil to draw a circle on the back of the slide. And remember not to place the bacteria on that same slide where you marked it. And then you take a sterile disposable loop, and remember to keep it sterile. And take a dropful of sterile water and place it in that drawn circle. And then with another sterile loop, you're going to get a bit of bacteria from a colony and place it in that water. So again, keeping that loop sterile, take just a little bit of a colony and touch it to the water. And then spread it around to like a thin dime-sized area. And then you're going to allow that smear to air dry completely, which takes about 5 minutes on a slide warmer. And then with quick movements, you're going to pass the slide over a flame about 3 to 5 times. And you should never hold it over the flame directly. And in the end, you'll see an opaque smear on the slide. Immediately, you'll drop a couple of drops of malachite green on the slide and let that sit for 10 minutes. Now remember that malachite green is staining the endospores. Then you're going to gently rinse that with water. And remember the malachite green will stain the endospores, but it will be rinsed off of the vegetative cells. So then you can counter stain the vegetative cells with safranin. So you would drop a few drops of safranin on there and let that sit for 45 seconds. And then again, you're going to rinse it, and then you will blot the slide dry with bibulous paper. And once that's dry, you'll be ready to observe it under a microscope. And you should see the vegetative cells stained pink, and the endospores will be stained green.